Or so, but you have reached the Time for Genetics genetic testing webinar for Mountain States Regional Genetics Network. I want to acknowledge our funders for this program, which is HRSA, and this is our funding statement. We are very appreciative to them for their grant funding, and if you have any other questions about this, you can see hrsa.gov. My name is Christy Weiss. I'm the projects manager for Mountain States Regional Genetics Network, and today I am joined by Michaela Perkins, our projects coordinator. Both of us are here to help you today behind the scenes with any technical issues, so please chat us if you need anything at all. I know we're all pretty familiar with Zoom, but just as a reminder, we are asking everyone to stay on mute during the webinar, and that is at the number 1 flag here. Feel free to turn on your camera if you feel comfortable or would like to. We are recording today for archival purposes, so please keep that in mind. This is an active participant list at number 3 and number 4. We are asking you to use the chat function for tech issues and Q&A, but we will ask you to hold off putting your question in the chat until we announce the Q&A section, because we're going to do something else with the chat today that's a little interactive. This webinar is part of the Time for Genetics initiative. It's a primary care, pediatric primary care education initiative across the 8 states in the Mountain States region. We are in our 2nd cohort, and these dots here on this map represent all the clinics that are in our cohort 1 and cohort 2. We hope that some of you are here today from that, but if you're interested in our pediatric clinic in our region, we are opening cohort number 3 on the applications for that are opening on July 1st. And here's a QR code you can scan if you would like to get some more information about our Time for Genetics program. Again, that cohort number 3 is opening on July 1st when we will be accepting new pediatric practices into our program. This statement here is from our creditors, Projects and Knowledge. It credits this for continuing medical education. It's a jointly accredited program. We will be offering today CME for physicians, PA CME for physician assistants, CNE for nurses, and CPE for pharmacists. And we'll give more information at the end on how to claim those credits. Our 2 presenters today, Daryl Scott and Haley Streff, do not have any disclosures for this presentation. So with that, it's my pleasure to welcome our speakers. Daryl Scott is coming to us from Baylor College of Medicine. He's a clinical geneticist at Texas Children's Hospital. And with him is Haley. And Haley is a genetic counselor at Baylor College of Medicine and Texas Children's Hospital. Today, they will be speaking on the topic, genetic testing. It's not rocket science. And with that, I'm going to stop sharing my screen and turn it over to Dr. Scott.
So this is an interesting topic. I love to talk about this because often people think this is such a complex thing. And to be honest with you, I teach the same thing to medical students. I teach this to residents. We teach this to everybody. And everybody says, wow, this is really all that hard. And the answer is, no, it's really not all that hard. And so we'd love to walk this through with you. And hopefully by the time we're done, you'll say the same thing. You'll say, you know what, this really isn't all that hard. So we really start off our evaluations in the same way every time, and you do the same thing in clinic. So our evaluations in genetics clinic will have like a medical history, family history, and physical exam. Ours might differ slightly from yours, and in that we put a little bit more time perhaps in the family history and maybe a little bit more detailed physical exam. But there's one important question by the time you get done with this. If you want to decide how to order genetic testing, the most important question is, do I know the diagnosis? In other words, after doing all this, are you pretty sure that this patient has this particular disorder? If so, then what you need is a triangle and a circle. And if the answer is no, you need 3 Gs. And we're going to talk about triangle, circle, and 3 Gs today. So let's first talk about this triangle. The triangle is a little bit different. It actually has a fat base, and then it kind of goes and peaks up to this top part. And we're going to divide it into 3 sections. But again, this is if you know what the diagnosis is, or you think you know what the diagnosis is. Each level has to do with how big the genetic problem is. When I say the genetic problem, not what the patient has, but like physically, what is the change in the DNA and how big is it? So on the very bottom level, we have chromosome aneuploidy, like adding a whole extra chromosome or taking a whole chromosome away. And also translocations, where pieces of chromosomes have been swapped. In this particular case, to detect those, you want a chromosome analysis. The next level is microdeletions. These are deletions and duplications that are so small that you can't see them with the naked eye. So you can't see it under a microscope, but are actually very, very important. In this particular case, when we're looking for these type of changes, we're looking for a microarray. And then the very last top part is sequencing. This is when the changes are on the order of just a few base pairs, or perhaps there's only one base pair or one letter in the DNA that is different. In this case, you want a sequencing test. And so these are the three levels that we use. And when we think about a genetic problem and say, what test should we get? We're thinking about how big is the change possibly going to be. So I'm just going to move all that to the side. And so again, we have chromosome analysis, microarray, and then sequencing. And I've also put down del-dup analysis. Deletion duplication analysis often comes with sequencing tests, and it's a way of detecting whether you have a piece that's perhaps a little bit bigger than sequencing can do. That piece might be missing. So for most of our genetic tests, we're interested in knowing if a single exon of the gene might be deleted or duplicated. That del-dup testing is something we almost always want because it gives us another level of understanding of what might be happening to that gene. So let's just start by just going through some examples. So in this particular case, you would look at this particular child and just based on your experience, perhaps even in the world, not just necessarily in the physician's office, you would know that this child has Down syndrome because of his classic facial features. If we're thinking about Down syndrome, we really want to get a chromosome analysis. The chromosome analysis is going to be able to tell us whether this child has trisomy 21 or 3 distinct copies of chromosome 21, or perhaps some other kind of change that gives him Down syndrome. How do we do a chromosome analysis? We often do this on a blood sample. We're particularly interested in the white blood cells in this case, because the white blood cells have DNA, have a nucleus, and you can grow them up in culture. And so we're going to get a chromosome analysis, and we're going to be getting these white blood cells that we're going to culture. A typical chromosome analysis starts out looking like this, just looking down the microscope, seeing all of these chromosomes that are all mixed up together. We then use a computer to help us align those in a way that we can then group them according to their number. Biggest number is number 1, all the way down to 21 and 22. This is what we call a karyotype. It's the actual picture. So sometimes people say, well, I'm going to order a karyotype. We usually say we're going to order a chromosome analysis, and then perhaps the karyotype, that picture, will be part of it. But the idea is that you're actually going to have a visual representation of what the chromosomes look like. This is the picture. When you say, well, how good is that picture? The resolution is about 5 million bases, or 5 megabases. How big is that? That's enough to put, let's say, like 50 genes into. If you have changes that are smaller than 5 megabases, you will not be able to see them. And also, there's sometimes changes that are even bigger that you may not be able to see, depending on how stretched out the chromosomes are on this analysis. But the maximum resolution is typically thought to be about 5 million bases. This will also catch chromosomal rearrangements, which is really important. Some of these chromosomal rearrangements can be very important as far as assigning a recurrence risk. For example, in this particular case, you see that there are 2 chromosome 21s that are actually hooked together on the tops. This type of chromosomal arrangement is very important and is something that's a possibility in Down syndrome. In this particular case, this arrangement can come with a recurrence risk that can be up to 100%. So if this rearrangement of 21-21 is found in a parent, all of their children will have Down syndrome. And so you can imagine that's quite different than if it's 3 distinct copies of chromosome 21. These are the type of changes that we want to check. If you see a child that has Down syndrome, the check that you want is a chromosome analysis. This is a different child, which you may or may not recognize, but has very classic features. This child has tetralogy of Fallot, hypocalcemia, a nasal voice, and also has intellectual disability. If you know the pattern, then it makes life easy, but it's possible that you do not. This pattern, it goes along with what's called DeGeorge syndrome. DeGeorge syndrome is caused by a deletion on chromosome 22q11.2. That's this deletion that's shown here. We use a microarray to be able to detect these changes. Why? Because they're so small that you cannot see them under a microscope. They're a micro deletion. And so we use a microarray, and here you can see the specific deletion in this patient is actually marked here. How does a microarray work? So a microarray is literally a glass slide that has these probes that have been printed on it. It's actually printed with a jet, like a jet inkjet printer. Remember those? Well, this time they're actually using that same type of technology to print DNA and add one nucleotide onto these little circles of DNA. It's about the size of a slide. It's about this big, and it has probes that are from different parts of the genome. So in this case, I've shown a top part of chromosome 1. It will have probes all the way along chromosome 1. And the density of that probes is actually far more dense than what I'm showing here. But the idea is you're going to probe each one of these pieces of DNA and say, how many copies of the DNA do we have there? They then use a laser scanner that costs more than my house to be able to scan these. And then you get a raw data that looks a little bit like this. So this is an actual picture of a microarray slide with 1 million different bases, 1 million different probes on it. If you blow it up, like literally just blow up the picture, you can actually begin to see that there's these little tiny circles. Those are the circles that are printed by that special printer, and each one represents a unique part of the DNA. There's also really possible you can see that there's some that are yellow, some that are red, and some that are green. I'm just going to replace this and just say, let's concentrate on these 3 spots. Depending on the color, it actually tells us whether there's 1 copy of the DNA there, 2 copies of the DNA there, or 3 copies. We would expect in most cases to have 2 copies of the DNA because you get 1 from mom and 1 from dad. But of course, if you have 1 that's missing, well, you're only left with 1, or you may have extra, like in the case of Down syndrome, you have 3 copies of all the different probes on chromosome 21. This information is then sent into a computer, and it gives us a readout that helps us to determine what pieces are missing or extra. And so in this case, we're looking at a piece of chromosome 1. This is the readout that shows a deletion on this piece of chromosome 1. If you blow that up, it actually shows you not only where the probes are, but the genes that are in that region. And so I can actually block off and say, this is exactly the part that's deleted. And then all those names, all these names in here, all the names of the different genes that are affected. Very helpful. The only thing that you can't see with a chromosome microarray is you can't see chromosomal rearrangements. So if there's a piece that's just been moved from one part of a chromosome to another, just swapped without a loss of DNA, you can't see those. And so there are limitations to this, and understanding that can make a difference in the choice of genetic tests that we choose for a specific question. Many of the people also have heard of fluorescent in situ hybridization, or FISH. And so whenever I give this talk, especially to medical students, they say, tell me about FISH. When do we use FISH? FISH technology is often used, especially prenatally, to do a very, very rapid check for things like Down syndrome or trisomy 18 or trisomy 13. It's a very limited test though, because all you get is a picture of a cell and it has certain spots in it. So in this case, if it was a cell from a child with Down syndrome and the chromosome 21 spots were red, well, this child would have an extra red spot. So he has 3 copies of chromosome 21. If on the other hand, this was a child that has DeGeorge syndrome, his FISH might look different. In this case, this is the probe for 22q11. In that case, you'd actually would miss or be deleted one of those copies of that FISH probe. And so this is good for checking things very rapidly, but you need to know what you're looking for. And it only checks 1 spot. So it doesn't help us completely with Down syndrome. It just says, yes, at least that one spot of chromosome 21 is extra. So when do we use this? It has these limitations of you can't tell the size of the deletion or duplication, and you can't tell its orientation, but it's very good for other things. So for example, we use it for speed. Sometimes in prenatal, we want to know very rapidly if someone has possibly Down syndrome. That would let us do that very quickly. Very quickly, meaning on the order, let's say 2 days, 48 hours. It also can give us a preliminary answer if we need to determine something quickly. For example, we don't like to take kids that have Turner syndrome and have hypoplastic left heart to surgery. They do not do well. And so if I expect or think that a child might have Turner syndrome and they're about to go to surgery tomorrow, I may run one of these tests to rapidly determine if that child may have Turner syndrome, in which case we would switch and not do that, and we'd explore other options for helping that child with their hypoplastic left heart. In the case of Down syndrome, we'd still need to run a chromosome analysis after we did FISH. And in the case of George, we're still going to have to run a microarray. And so again, it's speed and it gives you a preliminary answer. We also sometimes use this to confirm or to check family members. So let's say, for example, I find a child that has DiGeorge syndrome. I can rapidly check his parents to see if they also have this syndrome or his siblings. And so it makes life easy. But again, we're going to use these under special circumstances where speed is important or we already know kind of the answer to things. What about this particular diagnosis? This is a baby girl who has meconium ileus, and her brother has chronic lung disease. Again, if you're familiar with this disorder, it makes life very easy. If not, to tell you the truth, you could probably Google this and you'll find the answer. But many of you may know that this is cystic fibrosis. Meconium ileus is classic for this. What do we do for cystic fibrosis? What is the right test if we are suspecting this? In this case, the right test is sequencing with del-dup analysis. Why? Because most kids with cystic fibrosis have only small changes in their DNA, and specifically the gene CFTR that causes cystic fibrosis. This is another syndrome which you may or may not recognize. This case is a child that has profound hearing loss, but also has retinitis pigmentosa. Again, if you didn't recognize this, you could Google it and you would find the answer very rapidly. But some of you will know that this is a child then that has Usher syndrome. Usher syndrome, again, what would be the right test? It would be that sequencing del-dup analysis, because the changes in Usher syndrome tend to be relatively small, on the order of just a few base pairs usually. There is a difference though, because Usher syndrome is not just caused by one gene, it's actually caused by a number of different genes. And so instead of testing each gene by itself, we typically run what's called a gene panel. It's still a sequencing del-dup analysis, but it sometimes may be helpful to think about actually the top of this pyramid as looking just like a pyramid, like with blocks. In this case, that CFTR or cystic fibrosis gene is just one gene. You just get that one gene and that's all you need. On the other hand, since there's many genes that cause Usher syndrome, you're really looking to try to get a panel. And so there's specific panels for different disorders. That would be a great choice for Usher syndrome. Get an Usher syndrome panel that will check all the genes that are known to cause that disorder. What about this child? This is again a child who you might recognize based on the family history. It's a boy who has cognitive delay, but his mother has ovarian failure. His grandfather has ataxia and tremors. If you know the pattern, again, the diagnosis is very easy. And if you even knew this pattern and put it into Google, you'd find the pattern. But many of you may know that this is the pattern for Fragile X. And so Fragile X boys can also have mothers and grandparents who are affected with different disorders. Fragile X syndrome is something special. It needs something special because of the way that the actual changes is in the DNA. In this particular case, it's not one of those deletions or aneuploidy with an extra chromosome or even small changes in the DNA. Instead, it's a repeat problem. So this is the Fragile X gene, and in front of it, there's this area that has repeats. This is, let's say, a normal size repeat. This is a repeat that's a little too long, the same sequence over and over again. And this repeats very, very long. in this case, this gene, the normal one, will make a specific product that's important for brain development and function. If you get a little bit larger, it still makes that same protein, but it also makes a toxic product that's toxic to nerve tissue and also to ovaries. That's why you end up with problems with the neural problems, the tremors, the ataxia, and also the problems with ovarian failure. If the repeat gets big enough, you actually don't make this product. The body actually methylates this gene and stops it from working. So you don't make the product that's important for brain development. That's bad, and that causes Fragile X. You don't make the toxic product though. Based on the number of repeats, this one will be normal, 5 to 44. This one is called a pre-mutation, 55 to 200 repeats. It's going to have the ovarian insufficiency, the tremors, ataxia, those are all possible. And then this is a full mutation, usually 200 or more. And this is what gives you Fragile X. This is an unusual disease. Unusual diseases sometimes require unusual tests. So in this particular case, none of these repeats can be diagnosed using one of these normal tests on the triangle. Instead, what you need is a circle. So remember, we talked about a triangle and a circle if you know the disorder. In this particular case, what you need is what's called a repeat expansion test. It's an unusual thing, but again, if you know what the disease is and how the disease comes about, you know that this is the expansion test that you need. It's special. This child also needs a special test. This is a girl who has cognitive delay, seizures, happy face, but also has poor speech, poor walking, and happens to love water. One of the kids I saw recently that had Angelman syndrome was down at the beach. She was just happy as a lark, just loved it. These kids love running water. What does she have? She has Angelman syndrome. Angelman syndrome, again, has a specific test that's actually very powerful for it, and that's a methylation test because it's an imprinting problem. These are the unusual tests, unusual disorders that take a little bit of a different thing. So triangle for normal ones, circle for ones that are special. So let's just kind of go through this one more time. What's the most important question? Do you know the diagnosis? If you say yes, you need a triangle and a circle. And if you say no, then you need 3 Gs. What are those 3 Gs? So the first one is Google. You'd be amazed at how many diagnoses are made by Google. And the answer is we're on Google all the time. Why? Because we have thousands of syndromes that we're supposed to know about, right? It's hard to remember thousands of syndromes. And so we use Google to help us to both make diagnoses and also to help us know how to care for our patients. But let's say, for example, you have this patient. If this patient was to come into clinic, I would know immediately what he has based on what he looks like. But if you can slowly go through the pieces, you can find out too. So he has short stature, he has arched eyebrows, he has synophorus, which means his eyebrows kind of make a unibrow. It's got growth of hair right there in the middle. He has limb abnormalities you can't see here, but they can be like transverse, like almost like it's a limb that's cut off, or perhaps fingers that only several of the fingers are there. If you put all that into Google, like just short stature, arched eyebrows, synophorus, limb abnormalities, what do you get? This is what you get. And as it turns out, what does he have? He has what Google says he has. He has Cornelia-de-Lange syndrome. Google is powerful. That's the first one of your Gs. The second one is a geneticist. Because we're geneticists, we do this all the time. And so we may know patterns that you do not know or do not recognize. So for example, if this patient comes to see me and has seizures and intellectual disability, there's a lot of syndromes that can do that to you. But if his mother has large front teeth, well, that's a specific syndrome. This is Kbg syndrome. Yes, it can cause you to have very large front teeth, and mothers can be carriers of this and have children that are more intellectually affected and have seizures. It's a very unusual thing, but the pattern is known by geneticists, and we're like, yeah, we know that pattern. And so sometimes a geneticist can be very helpful. The third one is genome-wide. In other words, if you don't know what's going on, Google doesn't know what's going on, the geneticist doesn't know what's going on, you got to go wide. you got to like cover all the different genes and say, how can I then like look through everything to try to find the answer? Third G, genome wide. For example, if a child comes in and has developmental delay, intellectual disability, or autism, this is a huge part of the patients that come see me. But there's so many syndromes that cause this that I'm never going to find just one that's going to be the answer. I've got to go wide. I've got to be able to look at lots of different genes. So because there's thousands of genes that can cause this. So what do I do about that? Like I said, this is a very common problem in my genetics clinic. So I'll show you exactly how I explain this to parents. I'll say, hey, your child has developmental delay, intellectual disability, or autism. Why don't you come over here where we're going to do the exam? And I've got butcher paper that's on the exam table. I'll grab myself a pen and say, come sit down, let's draw some things out. I draw the same thing out every time. First of all, I draw out two large lines and I say, all the DNA can be drawn out symbolically like two large lines. I'm not like large lines, right? Symbolically. You get one copy of the DNA from mom. You get another copy of the DNA from the dad. The first thing we want to know is, are there any pieces, like big pieces that are missing or pieces that are extra? Because you should have two copies, right? But maybe you only have just one, or maybe your child has three. That would shift the balance of things. So the first test I want to run is one that can check for that. That test is a chromosomal microarray analysis. If that test is negative, well, then we'll go on to the next step. In the next step, we're going to look at all 20,000 genes at the same time. I select all the genes one shot. But specifically, we're going to look at the coding portions of those genes, the part that actually codes for the protein. But we're going to be looking for something as small as a single change in one gene. That single change in the gene can be found by what's called whole exome sequencing. This is the test. Now, as you can imagine, you're going to find a lot of changes in DNA. In fact, for every time we run this, we'll find 500 to 1,000 changes. But the lab's going to help me by taking those changes and saying, what changes do we see all the time in normal individuals or in many people in the population versus what are unique or rare? But they've got to be a change that possibly affects the gene, makes it not work. And also that gene's got to have some relationship to the problem that we're seeing. So in this case, it ought to be a brain kind of gene, a gene that affects brain development or function. The lab does this for me and returns to me a test result that will help me know if that specific change or what changes I should be thinking about as a possible cause for your child's intellectual disability, developmental delay, or autism. So what am I going to do? I'm going to go wide, look at all the DNA, look at all the genes. This is what I do. How often does this actually work? People ask this all the time. They say, well, how often do you make a diagnosis? If you said amongst all of the people who come to see us in clinic, we do make a diagnosis in a significant percentage. About 10 to 15% are going to be diagnosed by chromosomal microarray analysis, which is a good chunk. The other 25 or 35% will be diagnosed by whole exome sequencing. So all told, perhaps about 50% or about half the kids who see me in clinic, we're going to find a genetic diagnosis for. This is significantly better than it has been in the past. When I first started being a geneticist, it was like 5%. And we weren't seeing kids with developmental delay, intellectual disability. They didn't come see us because there was no way to ever find it. No one ever thought we'd get to the point where we could do this kind of stuff. But here we are. This is why genetics is so powerful today. So what are the take-home messages? Let's think about this. First, do I know the diagnosis? That's the big question. If so, what do you need? You need a triangle that's going to be bigger at the base, looking for big changes, medium changes, small changes. And based on what kind of change you're going to see, you're going to order that particular test. You might want to use FISH, but don't forget, FISH is mostly for emergencies. You want to know something fast. It helps you to confirm certain things, and it's good for chromosome changes and micro deletions and micro duplications. It cannot be used for anything smaller than that. So this is not useful for a sequence change. Then you also have the circle. That circle has special tests like repeat expansion tests or imprinting or methylation tests. The number of diseases that need this kind of thing are small, but they're important because some of them are ones that you may see. Probably again, the most common would be things like Fragile X, Angelman syndrome, Prader-Willi syndrome. There's only a certain list, but they're important to know. What if you can't do all this? What if that doesn't help? Well, then you got your 3Gs. If you don't know what this child has, then you're looking for Google, you're looking for a geneticist, or we're going to be going genome wide. That's how we get this done. This is the pattern for identifying what tests you should order. It's that simple. Again, don't forget, this is not hard. This is not rocket science. I'm going to turn the time over now to Haley to talk about how do you order that ideal test. And then we're going to come back and do what's called a chat check. In a chat check, we're going to actually run through various patients and rapidly say, what is the test that we want to order? And we're going to check and see if you've got the most important points that we're going to be talking about today. Haley, it's all yours.
Great, thank you. I'm going to switch over now. Okay, so Dr. Scott did a wonderful job kind of getting us to the step where now if you know what you want to order, how are we going to go about doing it? So I tried to come up with that important question. So again, just like, do we know the diagnosis? Yes or no? If you know how to send the genetic test, of course, go off and do that. We want you guys to be empowered to order testing for everyone. But if you don't, I struggle to come up with a nice 3G alliteration like Dr. Scott. So instead, I just have 4 steps. And so we're kind of going to run through those. Some tests only require 3 steps, but we're going to globally talk about them. And I'm going to give you an example about how we've implemented some of these at Texas Children's that really streamlined our workflow to increase genetic testing access for all of our patients. So first, lab selection. Where are you going to send this test to? You know what test you want now. Where is it going to go? So I'm going to recommend that you use one of many national genetic testing labs. There are some smaller labs that do more niche testing. So thinking about things like the Angelman syndrome example, not every single lab is going to offer Angelman syndrome, but these bigger labs that do testing nationally are going to have a very large test menu. And I strongly recommend you pick out 1 or 2 that you want to become very familiar with. They have a large menu and they can kind of be your go-to for almost any test you want to order. A lot of the tests you are going to be ordering, things like sequencing, microarray, Fragile X, and even exomes are going to be available at all of these labs. I'm not endorsing one lab over another, but some of the big ones in alphabetical order are Ambry, Baylor, GeneDx, Invitae, Lineagen, Prevention, and PerkinElmer. So you kind of think about trying to form a relationship with one of them. Almost all of them are going to have regional reps too. Some of them are genetic counselors, some of them are medical science liaisons. They're going to be very familiar with with their testing menu and can really guide you and get you plugged in. We have a local rep for one of those labs, and she has gone out to some of our community clinics for Texas Children's and has things like pre-filled out requisition forms and can work directly with the patients to facilitate sample collections. So these labs have lots of resources that we want you to take advantage of. But how are you going to pick which lab to do? One of the things I did want to highlight is that there are many of these large labs that have sponsored testing options. Typically, they're sponsored by a pharmaceutical company, often doing rare disease drug development. So if you were to send a sponsored test, you would have to help make sure that you consent your patient to have their de-identified genetic information provided to the third-party sponsor. The Heartland Regional Genetics Network has compiled a really good list of currently available active sponsored tests. So again, these are done at no cost. I know there's a lot of times a fear of a bill for a family or spotty insurance coverage, but if they agree to send that de-identified information, a sponsored test could be a really easy way to do this. There are sponsored tests for an array of indications. You can search this list by this indication, things like hearing loss, Duchenne muscular dystrophy, spinal muscular atrophy. Those tests are all included or sponsored somewhere. Other places to look at where to find labs, something that I use all of the time is Concert Genetics. You can create a free provider account there and you can search for any single gene or any type of symptom, and it will pull up any test domestically or any lab that will offer that test. So if you do the Google, you figure out your patient, you think they have Cornelia de Lange and you want to test for Cornelia de Lange, you go into Concert, type in Cornelia de Lange, and it will pop up with every lab that offers testing for that. You can compare labs side to side and pick the best test. Another good place to start is right here. The MSRGN actually has compiled a list of labs that do some billing or prior authorization work for providers. So that'd be a really good list to kind of start from. And again, pick 1 or 2 of those labs to try to form a relationship with to help you facilitate the testing process. The second thing you're going to think about, and I think is a big question mark, is billing. So I think the first two questions when you're looking at a lab is the first question, does this lab accept my state's Medicaid plan? Not every single lab is going to accept every state's Medicaid plan. So for instance, at Texas Children's, we've identified at least 2 labs that do accept Texas Medicaid. So we use those for a lot of our patients, both private and Medicaid patients, because we can really know the policies for one lab. You also should be checking to make sure they're in network with most private insurance companies. This should be readily available on websites. Almost all of these major labs that I'm referring to, or you're going to see on these lists, they should have really good billing websites, and they can very easily provide you a list of making sure that they're in network with all of these private payers. And also most will have a list of state Medicaid plans that they do accept. I think you also should consider and think about direct billing versus institutional billing. Direct billing meaning that the genetic testing lab is handling all of the billing components for you. Institutional bill or clinic bill is where you and your office are handling the billing. So that means you would be responsible for things like the prior authorization. The bill would come from your clinic as opposed to the bill coming from the genetic testing lab. We have done a ton of direct billing in our clinic, and it's really enhanced our uptake, and it's taken a lot of stress off of our staff because again, the genetic testing lab has taken on the burden of all of our billing. So like I said, when you're billing a lot of genetic tests, let's be real, almost all genetic tests are going to require prior authorization, especially if you have a private insurance plan. So again, this can be done by your clinic utilizing your own resources, but even better, it can be done by the genetic testing lab. So again, I'm referencing this list that was compiled of labs that do prior authorizations for you. So you basically send the lab all of the information that they require. In most cases, this is just what test you want to order after you use Dr. Scott's patterns to figure out what you want to order. And then you just need to send a justification. Sometimes this is a clinic note, and sometimes this is a letter of medical necessity. When you're thinking about prior authorizations, I'm going to go back and refer you to the Heartland Regional Genetics Network, who has a genetic testing toolkit. And so they already have compiled resources and have things like templates for letters of medical necessity, what to include in a prior authorization to really get you started. It has all of the things. Labs also offer this. I can think of more than one lab that on their billing website, they have things like a template for a letter of medical necessity, and they have checklists for what exactly they need from you to complete the prior authorization. And this also can usually always be submitted electronically through labs portals. So you're eliminating the back and forth of a fax, and you can get everything done in a portal and track it in real time. So then you need to order the test. We didn't touch on this too much, but most tests are going to be able to be completed on a buccal or a cheek swab sample. Almost any sequencing test and any of those deletion duplications, exomes, CMA, those can all be done on buccals, and they should be validated for buccal at almost any major commercial lab that you're working with. The exception to this rule is FISH and the chromosome analysis. Those tests, because of their testing methodology, always have to be done in blood. Almost any other test can be done on a buccal sample or saliva sample. Buccal samples are kind of standard of care, and they're used more commonly than a saliva sample. They're also easier to obtain for little kids who can't necessarily spit in a tube. But again, chromosome analysis and FISH analysis do have to be done on blood, unless it's a prenatal setting, then you can use things like an amnio fluid. And again, if you're working with a major genetic testing lab, you're going to be able to contact that lab or your regional representative and say, hey, I want to order a chromosome analysis on this patient. I submitted the order online, and that most labs will say, great, we will coordinate the blood draw for you. Some will have contracts with mobile phlebotomy labs. Some will have contracts with with national labs like Any Lab Test Now or a Quest Lab, and they will help that patient get an appointment and get their blood drawn. So again, labs have a lot of resources that you can use and should use to simplify this process. You should always be consenting for a genetic test as well. There's already been some work done with consent, and there's good consent resources online at the MSRGN as well. And then you can go ahead and send your test. So I make that sound a little bit easier, but just to kind of give you an example of real life. So at Texas Children's, in our genetics clinic, we were kind of forced to do this to some degree in 2020 because of the pandemic, and we needed to be able to coordinate more testing outside of our clinic. And we were also just We had reduction in staff, and I think we were struggling like many were. So we implemented a new workflow. So we use Epic in our clinic, but we look at the patient list, and one of our staff members before the clinic day starts, will add 3 asterisks to the notes for any patient seen that day. And those 3 asterisks indicate that that patient has Texas Medicaid. The genetic counselors, myself and my colleagues, we've identified 2 labs that we work with very regularly that accept Texas Medicaid. They also are in network with a majority of private payers. So we use these labs a lot. We're very familiar with their test menu and their policies, and we're comfortable using them. We also know that, again, they accept Texas Medicaid and they do not require prior authorization for any Texas Medicaid tests. This is information we just found out and learned about working with our regional representative from those labs. So we are able to order a same-day buccal sample for almost anything on the test menu for any of those patients with the 3 asterisks. So any of our providers can walk into a room and they might say, I know I need this test. And they go, oh my gosh, that's at one of these labs. I know I can order same-day testing because they have those 3 asterisks. Anyone who doesn't have those 3 asterisks, the provider automatically knows that we can't send same-day testing. And then we are lucky to have a staff member and a genetic counseling assistant who's able to submit all of the necessary materials through the lab's portals so they can complete benefits investigations and prior authorizations for the patient. Again, we're just giving them the information. We're not actually doing the prior authorization. The lab is. We're just providing the clinical information they require. The lab provides the outcome of that back to us, and then we are able to notify the patient so they can complete testing. In a vast majority of cases, we are just shipping buccal kits. And by when I say we are shipping buccal kits, I mean we are ordering a buccal kit to be mailed to the family through all of these labs portals. So we've really streamlined. Previously, I think when your staff is doing this, it's all on them to coordinate, follow up, check and make sure this has been done, re-notify patients, bring them back, send out things yourself. You don't have to take on a lot of that burden. And again, you can even work with your regional rep to streamline things to, like I said, have filled out requisitions, for example, or if you need a blood draw, they can coordinate the blood draw on your behalf. So just to kind of wrap up before we do our chat tech, I just wanted to bust a few myths. So I think there's people think that insurance doesn't cover genetic testing. And yes, insurance doesn't cover all genetic testing, but it has improved. For example, in the state of Texas this past fall, they added CMA as a covered benefit for all Texas Medicaid participants. So it took a while to get guaranteed coverage. Some managed Medicaid plans were covering it, but it's now a guaranteed benefit. So that's a great step in the right direction. And we have made a lot of strides. There are lots of payers. I am more familiar with insurance policies than I ever anticipated, but there's a lot of payers who do recognize the benefit of an early diagnosis, especially if you have a really good differential. If you say, here's why I think this patient has Fragile X, they're going to cover the Fragile X test because they recognize the benefit for that in the long run. I also just want to kind of re-emphasize what I've been saying, that these genetic testing labs have resources for you to use. They also want to make testing accessible to patients as much as they can. So use their resources. Again, I think it's helpful to just contact 1 or 2 labs, make a connection with them, become familiar with their policies. These large labs are going to have extensive test menus. And again, like in our clinic, we've pretty much narrowed it down to about 1 to 3 labs that we use on a very regular basis. And we're very familiar then also with the reporting policies and what they offer. There are even labs, for example, Invitae, they offer free follow-up genetic counseling appointments through Invitae for any patient with a positive test report. So if you were to order a panel test from them, it comes back positive, the patient can sign up for a free telehealth visit with the genetic counselor at Invitae. And that's at no additional charge to anyone, and it's coordinated directly with the family. So I think becoming familiar with some of these policies, again, there's not one that's better than the others. It's just a matter of their small nuances and just you learning about them and just trying to figure out those resources. Okay, so that is where I am going to stop. So I'm going to stop sharing again, and we'll let Dr. Scott hop back in for his chat check.
So this is actually the very best part of the whole thing. I'll just say we talk fast, don't we? The answer is we have a lot to cover in a very short period of time. It's funny because if a medical student was listening to this lecture, they'd listen to it at 1.5x speed. They'd actually make it faster so they could get to the stuff faster. You may want to listen to it at like, I don't know, depending on how old you are, you might want to do it 0.5, or maybe if you're my age, maybe 0.25, I don't know. Either way, we hope that you'll be able to capture the things that are the most important. Just kind of as a refresher of what we want to think about, we're going to go back to this stuff, okay? We're going to talk especially about the stuff that's on the top. So I'm going to talk about triangles and circles and how to pick the right genetic test. The way we're going to do this is kind of an interactive way. Is how do you be interactive on Zoom? And the answer is it takes a little bit of like work. But we can do this even in China. So I can sit here in Houston and talk to physicians in China and they can do this. So you can do this. It's really simple. We're going to give you a question. It's going to have a multiple choice answer. And we're asking you to put the answer that you think is correct in the chat, but don't push enter. We're going to give everybody 15 seconds or so to put their answer in. And then when we're all ready, you're going to say push enter. You're going to push enter. And on this other screen where I've got the chat pulled up, got two computers going, I'm going to see the answers that you're putting in. So if it's A, I'm going to see A, just blow up this chart. And you can see the same thing, right? And then we're going to go, yeah, like you guys are amazing. But if the answer is A and you get a D, I'm going to go, whoa, we needed to go over this a little bit more, right? And then we're going to solve each one of these so you can see exactly how we would walk through this in the clinic. So let's see what we get. Again, each one will have 15 minutes or 15 seconds, sorry, to do. And you'll look, and these are going to be the answers that we're going to pick. Single gene testing, panel testing, chromosomes, FISH, CMA, or then go first CMA and then do the whole exome sequencing. This is going to be it. So ready for our first question? Remember, put the answer in, but do not push enter until we are ready to go. So this is our first diagnosis or first patient that comes to see us. The question is, do you know this pattern? And if you know this pattern, what is the correct test to order? Again, we'll give you 15 seconds to put your answer into the chat. Do not push enter. And after 15 seconds, we're going to push enter. Okay. And I'll give you even a little bit of a countdown. So 5, 4, 3, 2, 1, enter. Okay, good deal. This is working. This is awesome. Okay, so how do we do this? Let's just walk through the way I would think about this. The first thing I guess I know is that I know these are cafe au lait spots, right? And I know those are tumors. And so just by looking at these two things, I can start to think about, wow, so what is the answer? If I've never seen this before, I would go to Google and put in cafe au lait tumor syndrome. By doing that, it actually pops up with this. And what do I get? Well, it looks like I got a lot of neurofibromatosis type 1. And the answer is, ta-da, that's the answer, right? Then the question is, what kind of change causes neurofibromatosis type 1? As it turns out, this is a really, really long gene, but it's only one gene that causes this. But that gene often has little small changes in it, or sometimes has a specific deletion of a couple exons. So in this particular case, I would say this is neurofibromatosis type 1. It's caused by changes in the NF1 gene. Because it's only one gene, I'm going to get single gene testing. So I'm like, yeah. If you said the same thing, good on you. If you said gene panel, it may be because you know that sometimes we test for NF1 and another gene called spread1 at the same time. That's true, but spread1 doesn't cause the tumors. And so you could actually do it with just NF1. If you said, hey, I had no idea what this was, well, then maybe you should have sent them the 3 Gs and sent them to us, because we'll know what this is and we'll get that done for you. We're all just here to help each other. But so far, so good. Ready for the next one? How about this one? This one, again, is a very classic thing that you might be able to say, I know what this is by just looking. This is a very special kind of tumors that are growing on this person's nose and cheeks. 15 seconds. Don't push enter. 5, 4, 3, 2, 1, enter. Okay, so got a little bit of a mixed bag in this one. Let's see how we do this. So first of all, what are these? These are little tiny baby tumors, right? And so I have an actual special name for them. This is Angiofibromas, and they're ash leaf spots, which are the white spots that are on the skin, and syndrome. If you put that in, you get these answers, which probably the easiest one is down here, although it's hidden in these other ones. But this is tubular sclerosis. If you recognize that, then the question is, how many genes cause tubular sclerosis? Again, we only give you 15 seconds, right? And so it's like, gotta like kind of know this. But if you say, well, this is tubular sclerosis, as it turns out, there's 2 genes for that. And so instead of doing a single gene, you're actually going to want to do a gene panel. And so we typically ask for a tubular sclerosis gene panel. This is going to do a great job on evaluating those 2 genes for you and giving you the answer in a great majority of patients that have tubular sclerosis. Doing so far so good? Not too hard, right? Not very much pressure here. Ready for another one? How about this one? What does this child have? 15 seconds. Okay, ready? 5, 4, 3, 2, 1, go. Yeah, all right. Okay, so what does this child have? First of all, I just look at some things. Did you notice the extra little digit on here? So this is polydactyly, has bilateral cleft lip and palate, also has just interesting eyes, forehead. This is a classic kind of look for a child. In this particular case, this is a trisomy 13 child. Like we would walk into the room and say, this is trisomy 13 until I prove otherwise. What would you pick? So I would pick a chromosome analysis because again, I'm interested not only in the 13, but to make sure that the 13 is not hooked on to something else. And so I'm going to say chromosome analysis. If you were to say something else, let's say fish, maybe you were trying to get this answered quickly because you need to have the answer now. I'm kind of a slow kind of guy, think through things. I'm going to have to get that chromosome analysis in the end. I'm willing to wait for a little while and make that happen. But again, that would be a reasonable one. Could this be identified by CMA? Yes, it could, but you're still going to have to go back and get a chromosome analysis because you need to know how the 13 is oriented. Is it stuck to something else? And so the sad thing is that sometimes we end up getting tests that are not ideal. In this case, the ideal test would be chromosome analysis. Okay, how about this child? Do you know what this is? Probably so. 15 seconds. Five, 4, 3, 2, 1, go. All right, a little bit of mixtures on this one. Let's go for it. So when I look at this, I think about the fact that this kid's got hearing loss. He's got a cochlear implant, which means he probably has severe to profound, more likely profound hearing loss on at least one side. The problem is that hearing loss can be caused by a whole bunch of different genes. Just non-syndromic hearing loss, if he has nothing else, that can be over 100 different genes. And so in this case, I can't do each gene by gene. There's no way. I'm going to need something that covers all those genes quickly. So I would say a gene panel. We have really good gene panels that do this, like really good. And they're going to find the great majority of the kids that have hearing loss. If that gene panel comes back negative, I can take a deep breath and say, okay, let's go for them for chromosome microarray and whole exome sequencing would be a reasonable thing, because then you're actually looking at perhaps the other 1,000 or so genes that can also cause hearing loss. But again, for a non-syndromic hearing loss, a gene panel would be really, really powerful. And then again, CMA and Wes, if that fails. All right, we'll do one more and then we'll call it. But this will be our last one, okay? 15 seconds for this one. 5, 4, 3, 2, 1, go. All right, again, a little bit of a mixture. Let's see what we've got here. In this particular case, he's got a very wide neck. Like, would you agree? That's a wide neck. If you just said, I don't know what this is, but I know wide neck. If you have wide neck syndrome, you're going to get it. In this case, this person has Noonan syndrome. Now you have to say, well, how many genes are caused Noonan syndrome? You see that each one of these is a little bit of a step, and you may not be able to know that immediately. That's okay. If I didn't give you 15 seconds, if I gave you 2 minutes, you get it. And so Noonan syndrome, how many genes cause Noonan syndrome? Last time I checked, 14. And so in this particular case, I want a gene panel. A Noonan gene panel would be the way we would do this. And chances are we're going to get that. If it's negative, what am I going to go for? Well, it wasn't what I thought it was, so I'm going to go for CMA and whole exome sequencing is what I'm going to do. All right, we're now going to go ahead and I'm going to shut this off. We've done our chat check, and we're going to open it up for questions.
And Dr. Scott, I had one that was private message to me. So while everybody's thinking and popping those in the chat, I'm going to read that if that's okay for you.
Sure.
I think this one actually might be a little bit more for Haley, but it says, when Haley was talking about the access to the 2 particular labs in Texas, do those apply to people of any age on Medicaid or only pediatric under 18? Or do you know if that's certain ages?
So, I mean, for us, so I work in a pediatric clinic, so that is Texas Medicaid, not Texas Medicare. So Texas Medicaid does extend, for instance, to some women like who are pregnant, for example. So there are exceptions to that, but it is that what I was discussing is Texas Medicaid. So that can include adults. It depends very much on your state Medicaid and Medicare rules.
Thanks for clarifying that. Appreciate it. And if anyone else has questions, please pop those in the chat. Also in the chat, you will see that we've put the link a couple times. Michaela has put that in there for our form for CME and CEUs, as well as our evaluation forms. So if you're not claiming CEUs today or a CME, please fill out that form and just give us some feedback if this was helpful for you, if the topic was helpful. But if you are claiming CME, we have to have that form in order to process that. So please take a look at that link in the chat. Are there any other questions? I want to thank you both. This has been incredibly insightful and helpful. And I just talked to a pediatrician today in our Time for Genetics who was asking for this specific type of information. So I hope it was helpful. I saw her pop on. She's viewing this today. So I hope this was helpful for all in attendance. Okay, we have a question in the chat there about what are the 2 labs in Texas that accept Medicaid. I don't know if you're at liberty to say that or not. Haley, that's up to you.
I mean, the 2 that we use that I'm aware of are GeneDx does and Invitae does. There may be more, but those are the 2 that I know accept Texas Medicaid.
Thank you for sharing that. And again, just a note, in today's talk, we are not endorsing any lab over another lab. I'm just trying to provide names of ones to get people started, that there are quite a few to choose from out there. And I did get a direct message about offering CEUs for social workers. And at this time, we do not have CEUs for social workers. I do apologize. Today, it is CME for doctors, nurses, and then physician assistants and pharmacists. All right, another question here popped up. Do Texas Medicaid STAR plans require prior authorization?
So in general, yes, like I kind of alluded to, there are some, like for instance, some of the labs we work with, they don't require like that we complete a prior authorization before sending the test. But like, yes, in general, all genetic tests for Texas Medicaid require prior authorization.
Great, thank you. And I think this one might be for Dr. Scott. So say a West test discovers a variant of unknown significance, unknown variant, a rare gene mutation. Any way to quickly check if there are any clinical or preclinical studies interested in possibly following up further?
This one's a little bit of a challenge. And so the first thing we would usually do is we actually would say, I want to know the inheritance of that particular variant. And so sometimes the very first start is to clarify what's happening within the family. If it's a de novo variant and you're still not sure of its cause, you can connect with other people who are interested in that gene using a thing called Gene Matcher. And so we routinely go on to Gene Matcher. You can set up a free account, type the name of the gene in, and it will hook you up or send you the emails of all the people who have registered interest in that gene. Sometimes there's individuals who can actually do functional studies on that gene, or you may also look up the person who is actually kind of the preeminent person in the world on that, and using another portal called Gene Reviews. They're often written by experts who may be able to put you in contact with someone who has a functional test. I'll just tell you personally, I wish there were a lot more functional tests, but often we are stuck with not knowing for sure whether a variant is of importance or not. That's why at least knowing how it's inherited in the family is often the first and most important step.
Thank you so much. And I see one more question. I know we're at the top of the hour, so do you have time for one more?
We do.
This is for Dr. Scott. Could you speak to the usefulness of programs like Face2Gene or Haley?
So Face2Gene is a helpful thing, especially for researchers. And we like to see if there's specific features that are important for a disease or a specific disorder. There are limitations to being able to do so. Your eyes are very, very good. Like your eyes are better than you think. And so sometimes you should just be calm and just be able to say, well, what does this child have? Is it normal or is it abnormal? What about their eyes? What about their ears, nose, mouth? And if you can put those down into words, that's powerful. Some of the limitations of Face2Gene is that there's a lot of variation in some syndromes. And so sometimes we only see a fraction of the features. You know, if a child has 50% of the features of a syndrome, that might be incredible. And so there are limitations. There are also things where there's variability within some syndromes. For example, some of the deletion syndromes can be bigger or smaller or even in different parts of the DNA. In that particular case, it's very important to know what part of the DNA or how exactly was that face determined that makes the Face2Gene profile. So we find them interesting. But actually in the clinic, I tend to just use my own eyes.
Great, thank you so much for that. I don't see any other questions popping up, but so I think we can end here. And I just want to give you all a round of applause, virtual round of applause. And thank you so much for your time today and your preparation. And the chat check was amazing. So thank you for that extra engagement today. So with that, we'll sign off, and we have these webinars quarterly. Our next topic area will be on autism in September, so be watching for that. If you are not connected with Mountain States, please feel free to fill out the form I just put in the chat to reach out to us with any other further questions. And again, big thank you to Dr. Scott and Haley today for their time and efforts.
You've had a real pleasure.
Thanks again.
Bye.
Bye-bye. You're getting some thumbs up there. Bye-bye.